24-year-old man is brought to the emergency department and requires urgent endotracheal intubation. He is given succinylcholine to facilitate rapid muscle relaxation. The intubation is successful and the procedure appears uneventful. However, even 30 minutes later, the patient remains flaccid and apneic, unable to breathe spontaneously, and requires continued mechanical ventilation. On further questioning, his family reveals that his brother experienced a similar episode of prolonged paralysis after surgery in the past. Take a moment, think about the mechanism of acetylcholine transmission, and comment your answer in the comment section below. And by the end of this video, you will not just know the answer, you will understand why every other option is wrong. In our previous videos on the autonomic nervous system, we first built the foundation. We understood what the autonomic nervous system is. Then we discussed the thoracolumbar and craniosacral outflow. After that, we went deeper into the neurotransmitters involved in both systems and the different receptors. Now that the basics are clear, in today's video, we are going to focus specifically on an important part of the parasympathetic system, that is, the cholinergic system. More precisely, we will understand cholinergic transmission, starting from how acetylcholine is synthesized, how it is handled inside the nerve terminal, and why each step is clinically important. So let's get started. Acetylcholine is synthesized inside the cholinergic nerve terminal. First of all, choline has to enter the nerve ending, and it does so through a carrier-mediated transport system. In fact, this is the rate-limiting step of the entire process. Once choline is inside the neuron, it combines with acetyl-CoA, which is already present in the nerve terminal and is derived from the mitochondria. This combination is catalyzed by a specific enzyme called Choline Acetyltransferase. As a result of this enzymatic reaction, acetylcholine is formed. Immediately after it is synthesized, acetylcholine is not left free in the cytoplasm. Instead, it is packaged and stored inside small synaptic vesicles. When the nerve impulse or action potential travels down the axon and finally reaches the nerve terminal, as soon as it arrives at the presynaptic membrane, where the electrical signal causes voltage-gated calcium channels to open. Consequently, calcium ions rush into the nerve terminal because the calcium concentration is much higher outside the cell than inside. In response to this calcium entry, the synaptic vesicles that contain acetylcholine move toward the presynaptic membrane and fuse with it. As a result, acetylcholine is released into the synaptic cleft by a process called exocytosis. After being released, acetylcholine diffuses across the synaptic cleft and binds to cholinergic receptors on the postsynaptic membrane, that is, muscarinic and nicotinic receptors, thereby producing its effect. However, this action is very short-lived because acetylcholine is rapidly broken down by the enzyme acetylcholinesterase. So in summary, the arrival of the nerve impulse opens calcium channels, calcium enters, vesicles fuse with the membrane, and acetylcholine is released into the synapse to transmit the signal. This acetylcholine is hydrolyzed to choline and acetic acid by the enzyme cholinesterase, and this breakdown terminates the action of acetylcholine within milliseconds. There are two types of cholinesterases: true cholinesterase and pseudo-cholinesterase. First of all, true cholinesterase, also called acetylcholinesterase, is present at the synapse, that is, at cholinergic nerve endings, autonomic ganglia, and most importantly, at the neuromuscular junction. Therefore, its primary role is to rapidly destroy acetylcholine right at the site of action. Because of this, it directly controls the duration of neurotransmission. Clinically, this is highly important because drugs like neostigmine, physostigmine, and organophosphates inhibit true cholinesterase, leading to accumulation of acetylcholine and exaggerated cholinergic effects. On the other hand, pseudocholinesterase, also known as butyryl cholinesterase, is present mainly in plasma, liver, and other tissues. Unlike true cholinesterase, it is not primarily responsible for synaptic transmission. Instead, it hydrolyzes certain ester drugs in plasma, most importantly succinylcholine, which is a neuromuscular blocker, and some local anesthetics like procaine. So patients with pseudocholinesterase deficiency cannot metabolize succinylcholine properly, and as a result, they develop prolonged apnea after anesthesia. Acetylcholine is hydrolyzed by both true cholinesterase at the synapse and pseudocholinesterase in plasma, rapidly by true cholinesterase and relatively slowly by pseudocholinesterase. If we administer acetylcholine intravenously, the moment it enters the bloodstream, it is rapidly hydrolyzed by pseudocholinesterase present in plasma. So a large portion of the drug is destroyed before it even reaches its target tissues. Now, whatever tiny fraction of acetylcholine survives in circulation and reaches a synapse faces another problem. At the synaptic cleft, true cholinesterase is present in very high concentration, and it hydrolyzes the remaining acetylcholine within microseconds. Therefore, at the site of action, acetylcholine cannot persist long enough to produce a sustained effect. So effectively, acetylcholine is destroyed at two levels, first in plasma and then in the synapse. Because of this rapid breakdown, its duration of action is ultra short and it cannot be used clinically as a systemic drug. So to overcome this problem, scientists modified the structure of acetylcholine and developed methacholine. The idea was simple. If acetylcholine is being destroyed too quickly, then we need a molecule that looks similar enough to stimulate the same receptor but slightly different so that the enzymes cannot break it down so rapidly. Therefore, a small structural modification was introduced. Specifically, a methyl group was added to the acetylcholine molecule. Because of this methyl group, methacholine becomes resistant to plasma pseudocholinesterase. As a result, when methacholine is administered, it is not rapidly destroyed in the bloodstream like acetylcholine. However, when methacholine reaches the synaptic cleft, it can still be hydrolyzed by true cholinesterase at the synapse, but at a slower rate compared to acetylcholine. This is actually beneficial because it prevents the drug from having an excessively prolonged or toxic action. So compared to acetylcholine, methacholine has a longer duration of action, especially at peripheral muscarinic receptors. That is why it can be used clinically. Most importantly, in the bronchial challenge test to diagnose asthma, where it induces bronchospasm or bronchoconstriction in hyperresponsive airways, which we will discuss in upcoming lectures. Now, as we move forward, I would like to clarify a very important concept that we frequently use in the ANS, and that is the difference between direct acting and indirect acting drugs. Suppose this is a drug, and a drug that binds directly to cholinergic receptors, either muscarinic or nicotinic receptors, and accordingly activates or blocks them. Such a drug is called a direct acting drug, as it works directly on nicotinic and muscarinic receptors. Does it require the presence of a nerve terminal and acetylcholine to do its job? Not at all. These drugs themselves interact with the receptor and produce an effect by modulating receptor activity. So the drug directly mimics the action of acetylcholine at that receptor site. And in contrast, if a drug is not working on muscarinic or nicotinic receptors, but rather is working on the nerve terminal or in the synapse, working on this big blue box, then this drug is said to be an indirect acting drug. So in this case, the drug acts somewhere else in the pathway of acetylcholine transmission, but the drug does not bind to the receptor by itself. The only way the drug causes responses in the patient is by changing the levels of acetylcholine indirectly. The most important example of an indirect acting drug is acetylcholinesterase inhibitors. They increase the level of acetylcholine in the synapse by inhibiting acetylcholinesterase. As a result, more acetylcholine accumulates in the synaptic cleft, and this excess acetylcholine then stimulates the receptors. If there is no synapse or nerve terminal present, an indirect acting drug does not have any effect on the receptors. Now that we understand what indirect acting drugs are, let us discuss a few drugs that we study nowhere in the ANS but only here. The first one is Hemicholinium. Hemicholinium is an indirect acting drug that works at a very early step in acetylcholine synthesis. It blocks the uptake of choline into the nerve terminal. Now think logically about the consequence. If choline cannot enter the neuron, then it cannot combine with acetyl-CoA. As a result, acetylcholine will not synthesized, and if acetylcholine cannot be synthesized, then over time the stored vesicles become depleted. As a result, when a nerve impulse arrives, less acetylcholine is available for release. So the ultimate effect of hemicholinium is a gradual reduction in acetylcholine levels at the synapse, leading to decreased cholinergic transmission. It is important to understand that this effect is not immediate. Initially, the already stored acetylcholine can still be released. However, as the stores get exhausted and new synthesis is blocked, neurotransmission becomes progressively weaker. Clinically, hemicholinium is not used therapeutically because of its toxicity. The next one is Vesamicol. Vesamicol is an experimental drug. It does not block choline uptake like hemicholinium. Instead, it blocks the vesicular acetylcholine transporter. Now remember, once acetylcholine is synthesized in the cytoplasm, it must be transported into synaptic vesicles for storage. Vesamicol prevents this packaging step. As a result, acetylcholine may still be synthesized, but it cannot be properly stored inside vesicles. Consequently, when a nerve impulse arrives, there is little or no acetylcholine available for release, and over time, this leads to depletion of functional neurotransmitter stores and reduced cholinergic transmission. Now there is another important indirect mechanism, and that involves botulinum toxin. It doesn't affect synthesis or storage directly. Instead, it blocks the release of acetylcholine by preventing the fusion of synaptic vesicles with the presynaptic membrane. It does this by cleaving SNARE proteins, which are necessary for vesicle docking and exocytosis. So even though acetylcholine is synthesized and stored normally, it cannot be released into the synaptic cleft. In exams, botulinum toxin is commonly tested in two ways. First, by its side effects, and second, by its therapeutic uses. Now first, how does a person get this toxin? Botulinum toxin is produced by the bacterium Clostridium botulinum, which is an anaerobic, spore-forming organism. A person can get exposed to this toxin mainly in three ways. First, by foodborne. This happens after eating improperly canned or preserved food where the bacteria have produced the toxin. The toxin is already formed in the food, so when the person eats it, the toxin directly enters the body. Second, this occurs when an infant ingests spores, commonly from honey. The spores germinate inside the intestine and produce toxin locally. And lastly, spores contaminate a wound, germinate, and produce toxin. And now, what are the consequences of this toxicity? Botulinum toxin decreases acetylcholine levels at both muscarinic and nicotinic receptors. So technically, any cholinergic synapse can be affected, whether it is muscarinic or nicotinic. However, the major concern is at the neuromuscular junction of skeletal muscle, where nicotinic receptors are present. Why? Because skeletal muscle contraction depends entirely on acetylcholine release. When botulinum toxin prevents acetylcholine release, the muscle cannot depolarize and therefore it becomes weak and eventually paralyzed. Although muscarinic synapses are also affected, which can lead to symptoms like dry mouth or decreased secretions, the life-threatening complication comes from paralysis of respiratory muscles due to blockade at the skeletal muscle junction. Now, regarding the use of botulinum toxin, interestingly, this toxin is used in controlled doses for medical and cosmetic purposes. So, a local injection of botulinum toxin reduces acetylcholine release, leading to muscle relaxation or paralysis at nicotinic receptors and decreased secretion at muscarinic receptors. Because of this property of causing localized muscle relaxation, botulinum toxin becomes very useful in conditions where there is excessive or involuntary muscle contraction. One classic example is blepharospasm, which refers to involuntary repetitive contractions of the eyelid muscles. In blepharospasm, the orbicularis oculi muscle contracts abnormally, leading to uncontrollable blinking or even sustained closure of the eyelids. Now, when botulinum toxin is injected locally into the affected muscle, it blocks acetylcholine release at the neuromuscular junction. As a result, the overactive muscle relaxes and the involuntary contractions decrease significantly. It's also useful in cases of dystonia. Dystonia is a movement disorder characterized by sustained or repetitive involuntary muscle contractions. These contractions can cause abnormal postures, twisting movements, or painful muscle spasms. The problem is not muscle weakness. The problem is excessive uncontrolled muscle activity. Therefore, Botox works by blocking the release of acetylcholine at the neuromuscular junction. The next condition is strabismus. It is a condition where the two eyes are not properly aligned because one or more extraocular muscles are overactive or pulling more strongly than the opposing muscle. As a result, one eye may turn upward, inward, outward, or downward. Botulinum toxin, when injected into the overactive extraocular muscle, that muscle becomes temporarily weakened or relaxed. In cosmetics, Botox is mainly used to reduce facial wrinkles, especially dynamic wrinkles, the ones that appear when you move your face, such as frown lines, crow's feet, and forehead lines. Next is hyperhidrosis. Hyperhidrosis is excessive sweating, especially on palms, soles, axillae, and face. Sweat glands are stimulated by the sympathetic nervous system, but, and this is very important, the neurotransmitter acting on sweat glands is acetylcholine, not noradrenaline, as we already discussed in previous lecture. And these glands have muscarinic receptors. So botulinum toxin blocks the release of acetylcholine from the sympathetic cholinergic nerve endings supplying those sweat glands. After botulinum toxin, aminoglycoside antibiotics such as Gentamicin can also impair calcium-dependent acetylcholine release. Clinically, they can worsen neuromuscular weakness, especially in patients with myasthenia gravis. High levels of magnesium can also inhibit calcium entry into the nerve terminal. Magnesium acts as a calcium channel antagonist. Because calcium and magnesium have similar charges, magnesium can compete for the same entry points. So when magnesium levels are excessively high, less calcium enters the terminal, and as a result, the signal to release acetylcholine is muffled, leading to a significant drop in neurotransmitter release. This is why excessive magnesium can cause muscle weakness. In the last example, the black widow spider also affects neurotransmitter release. But interestingly, it does so in the opposite way compared to botulinum toxin. The venom of the black widow spider contains a toxin called alpha-latrotoxin. Instead of blocking acetylcholine release, this toxin causes a massive, uncontrolled release of neurotransmitters from the presynaptic nerve terminal. It acts on presynaptic proteins and forms pores that allow excessive calcium influx. And we already know calcium entry is the key trigger for vesicle fusion and neurotransmitter release.